All right. I want to be respectful of everybody's time. I know a lot of people kind of jump in on their lunch hour if you're on the East Coast. And good morning to those of you on the West Coast and everybody in between. Happy daylight savings. We were just rejoicing the mental health benefits of having that extra hour in the evening to get your walk in or whatever you need to do to compress, because these are strange times. I just want to say that this is, or in January, we celebrated 1 year of this seminar series. And really the goal of this is to create a home, a community, a watering hole for both leading and emerging scholars in women's brain health space. And what I mean by that are, when we think about women's brain health, because really this should just be brain health without the modifier. But it's really trying to address the fact that for so long brain imagers have had these incredible tools to peer into the brain and understand how it works. And yet, thanks to some incredible scholarship by Laura Pritchett, who I think is on this call, we now have an understanding of just how little those tools have been applied to conditions or experiences that affect women either uniquely or differentially or just differently. So we have a lot of work to do to fill this space. And this is hopefully a way to kind of create a home for leaders that have been doing this good work for so long, but too often do it in isolation. So this is like our baby step to just create a home for all of us to be able to lean on one another and learn from one another. I think those of you in this space have recognized that women's health is sort of having a moment. Hopefully it's not a moment, but a movement. We've had special issues in Nature, Science Advances last week. There's more to come. There's even this recognition beyond academia that investing in women's health cannot wait. World Economic Forum, this is like a huge issue for them right now. At the next forum, they're going to have a special on women's brain health. They had a report in January suggesting that filling the gender data gap across the biomedical sciences would unleash about a $1 trillion return on investment. So, as I like to say, like whether you're driven by the moral imperative or by cold hard economic facts, investing in women's health makes sense. And suddenly the world is waking up to that. So it is a great time to be entering this space as both trainees and senior researchers. The last administration held the first ever White House conference on women's health research. They were excited to kind of put a lot of money behind that. Today is a new day, right? The last few months, I'd say that heyday has turned into some very strong headwinds. We have an administration that has labeled women and female as flagged words in grant applications. Like the 180 could not be stark enough. And so one, I just want to address the fact that these are weird times, not just for women's health, obviously. University and medical centers are confronting a challenge that in our lifetimes we have not felt before. And I think people are feeling very fragile. So thank you especially for coming together. And I hope that this can be a home to help us navigate through some pretty uncertain times. And nobody knows what's going to happen, but hopefully we can try to navigate it together. That's all I'll say. But I hope as you're sort of making this argument to people in your lives, that when we think about things like perimenopausal brain fog or, peripartum depression, like these are issues that transcend politics, right? These are issues that don't care whether you wear red or blue or what side of the aisle you sit on. And hopefully we have an administration that can recognize that. But there's a lot going on besides just the attack on academic centers, and that can be very stressful too. So just want to acknowledge that. Okay, enough for me. Let's get on to the science. There's so much to say, and maybe we could just have one big therapy session together.
I like that
let's
idea.
get on to what makes us all very happy. So this is a roundtable discussion. This is the first time we've held one of these, addressing best practices and emerging opportunities in human brain imaging and how they can be applied to the study of women's brain health. So we have some leading experts here that are just fabulous methodologists and theoreticians, and we want to pick your brain and see how we can apply the incredible work that you're doing with a sort of specific lens to women's health. So I'll be quiet and let the real sort of brilliant folks on this call take the stage, but thank you for coming.
Thank you so much, Emily. It's always nice to hear some inspirational words when we're all feeling a little down. At least I am. My name is Kuceyeski. I'm a professor in the radiology department at Weill Cornell Medicine and also at Cornell Ithaca in the computational biology department. And I've been working with Emily for a long time now on different sorts of data sets. We'll get into that later, but first, I want to have everybody go around and introduce themselves briefly. Tell us your sort of title, your institution, and your main area of interest. And then, of course, Nina. Nina and I will be taking turns asking questions. Yeah.
Hi everyone, I'm Nina Miolane. I'll be joining Amy in asking questions today. I'm an assistant professor at UC Santa Barbara and with Amy, co-director of AI for the Bowers Women's Brain Health Initiative. So I work in AI for brain imaging and fundamental and clinical neuroscience. All right, next, Ted, maybe.
Hey, I'm Ted Satterthwaite. I'm at the University of Pennsylvania. My lab mainly focuses on brain development during adolescence, but also we have a big focus on tools for reproducible neuroscience, so image processing pipelines, open data, and open science.
Thank
Great,
you,
Catie.
Catie.
Yep. Oh, sorry.
Hi, I'm an assistant professor at Vanderbilt University in the Department of Electrical and Computer Engineering, and our work focuses on methods for analyzing fMRI data to study the dynamics of brain activity, as well as integrating fMRI with other modalities such as EEG.
And Monica? Oh, sorry. Monica, can you hear me? Oh, yep.
Yes, sorry, I'm
Perfect.
having some Wi-Fi problems.
Oh, no.
Ironically, I'm in my office, so I'm going to
Okay.
switch, but yes. I'm Monica Rosenberg. I'm an associate professor in the Department of Psychology and the Neuroscience Institute and the Institute for Mind and Biology at the University of Chicago. My lab builds models that predict individual differences in cognitive and attentional abilities, and also tries to use modeling approaches to understand how those processes change over time in the same subject on timescales all the way from 1 minute to the next to across development.
Awesome, great. So we had another panelist who unfortunately couldn't attend, Joshua Buckholtz. They had a family emergency. So we will go ahead and start. So this is to everyone. We'll take turns. What do you think are the biggest roadblocks and the biggest potential opportunities for computational neuroimaging applied to women's health? So I guess since I'm talking, I'll go first. The biggest roadblocks, and I think WBHI is going a long way for this, is the lack of data. So I think we don't have a lot of women's health data out there. We have like a few 28andMe, we have the pregnancy data, we have some HCP aging data that has hormones, sort of, but like the overall sort of roadblock, I think, is the lack of data. So I think that's the sort of biggest thing for me. And Emily and WBHI is going a long way for collecting that. So I'm really excited about that in the coming years. And I think the biggest potential opportunities, I think, are if we can get those data, then we can do some really cool science, right? So we'll talk about some of it today, but looking at how sort of the anatomy and physiology of the brain changes with these life events that are specific to female bodies. So I'll stop there.
I'd like to jump on that because, yeah, I completely agree that the lack of data is the biggest problem. And especially for Amy and I working in AI, I'd like to recognize that the data we have in neuroimaging and in women's neuroimaging in particular is so small compared to what AI is used to. So AI wants images like tens of millions of images in a data set to perform reasonably well. And we are 3 orders of magnitude below that. So how can AI help foster revolution in women's health if we don't have the data? Which leads me to the opportunities beyond, the women's health discoveries that will come if we have more data. We can also ask, could we invest in the AI methodology to develop AI methodology that can work well with less data? So that's kind of one area of research that I'm interested in. Can we develop AI model that work as well with 1,000 times less data?
Ted, do you want to address that question?
Catie, go ahead. You unmuted us.
Sure, I can just add a bit more to that as well, which this is a great point, and I feel that from what I've seen in the space of women's health, the type of data that needs to be gathered is so heterogeneous. So anything from, hormone levels to structural and functional brain imaging. And so ensuring that we have rich enough data both within and between individuals to be able to model these effects also seems like a challenge, but also an opportunity given the machine learning and modeling tools that are being developed.
And I'll just add one more thing, which is I'm not plugging WBHI in any selfish way, but, but I will say that they are working on standardizing a questionnaire that is like what you need for machine learning, right? Like you need some kind of standardized data collection in order to make it very easy to apply algorithms to it. So who wants to go next?
Yeah, I think I don't have a lot to add. I think more data, absolutely, because in both brain development and also transitions and across women's lifespan, there's just a ton of heterogeneity, and we're not going to be able to understand it with small n samples. I think that's what's exciting about WBHI is there's going to be a lot of data generated, but I think the other part of it is that just like we need smart people working on this problem. It's just been a historically incredibly understudied field. So we need more smart brains looking at those brains that we're going to be collecting. And that's, I think that it's like the moment and hopefully movement that Emily is referring to, hopefully that's changing.
Everybody listening, he's talking to you. All right, Monica.
I totally agree. And I think, I mean, in many questions, we're not like data limited, we're theory limited, but I really think we are data limited here. And I also, want to point out from the sort of methods analytical standpoint, we know that brain-based models that predict individual differences in behavior, changes in behavior over short and long timescales, don't work the same for everyone. And I think we need to take that into account when building and validating these models. If we have a model of a cognitive process or a behavior outcome or a symptom trajectory, we shouldn't expect that, the same model applied to everyone will be equally informative, but perhaps start pursuing these models that are informed by other aspects of when the data were collected, right? When in a person's lifespan, when in, a particular time of day, time of year, et cetera. And then aspects of the person, like their sex and gender. And I think we know those things matter for this sort of prediction, but I've yet to see it proliferate in the way that I think it can in the future.
Thank you. I love that you mentioned brain behavior because that's a perfect transition to our next question, which is for Amy. So what is brain behavior mapping and how can we use it to understand women's brains?
So it was perfect, Ma.
Yeah, thank you.
So brain behavior mapping is exactly what she just said, where you take a picture, the neuroimaging, you extract some metrics, whether it be connectivity, structural connectivity, functional connectivity, cortical thicknesses, and you map those to some kind of outside of the scanner behavior that's collected. And I wanted to follow up on what Monica said, which is that the heterogeneity in the data is something that's often addressed, and the heterogeneity of like the imaging and the heterogeneity of the behavioral measures is something that we all know exists, but the heterogeneity in the mapping is not something that's commonly done. And so we have something that just came out. I think it's in I can't remember where it is, biological psychiatry somewhere, global health, global something. But anyway, I could point you all to it. But it's basically looking at how we can model the heterogeneity in the mapping, right? So there might be some ways that we can incorporate a demographic information, whether the person's sex or gender or age, that would inform how the mapping happens, right? So we've done this in a very, very like easy way, which is just training models on males, just training models on females, and sort of testing them on the opposite, right? So just to see if the mapping is the same between brain and behavior for male and females. We've seen very similar things for, let's say, cognitive scores in healthy control populations. The male and female models seem to do equally well, but other things tend to have females mapping more with a higher level of accuracy. So things like ADHD, we've seen the models that we train on females that tend to perform better for females, but also better for males, which is interesting. And the same goes for things like predicting stroke outcomes. So it seems like the stroke outcome models that we're training on female data tend to have much higher accuracy than the ones trained on male models. So we're still trying to figure out why this is the case, but it could be that the neural substrates that underlie the behavior vary between the sexes, right? So this is something that we should be sort of exploring when we're talking about brain behavior mapping and women's brain health. Oh, and then let's see if anybody has anything to say, just kind of interject. But the next question we have is for Ted. So your group has led these large-scale efforts to map brain maturation early in life, and its aims are to shed light on typical and atypical patterns of brain development. Can the methodological and theoretical insights gleaned from developmental studies be applied to aging trajectories later in life, for example, in the context of menopause? And how can researchers get a more mechanistic handle on how major endocrine transitions, including puberty and menopause, influence these brain trajectories? It's a lot of questions, but if you forget,
Awesome.
just tell me.
I'm going to answer every one of those for sure. So yes, much of our work is focused on brain development in adolescence using multimodal neuroimaging, with really the goal of understanding, well, what is normal adolescence, including puberty, and how does variation and how kids like navigate that transition associated with things that we care about, like cognition or psychiatric symptoms. And I think the analogy to endocrine transitions in women's lifespan is like very clear. So puberty is a major endocrine transition, so is pregnancy, so is menopause. And we really should think of these as critical windows of brain maturation. And just like we study adolescence as a period of neuroplasticity, that framework probably is a really valuable framework for studying pregnancy and menopause. And like plasticity is like a very well-developed basic literature behind it from animal literature. And so like kind of taking that framework and applying it to a different phase of life that's historically radically understudied, I think could be really powerful. And one thing that we like know from adolescence that I think could be like kind of applied very easily is that like we know that the brain develops differently at different part of the brain. So a basic question we could ask in pregnancy and menopause is where and when do we see these brain changes in response to major endocrine shifts? It's like super simple, but relatively undefined still. And these sort of questions really require that we understand what is quote unquote normal. Like what is like, and we know like people are are quite variable. Everyone's got a different brain. But like looking across the population, what is the range of variability in terms of both where and when? And then once we have that information, can we unpack it to really understand why are some women particularly vulnerable to symptoms as they navigate menopause, and why are some women more resilient? I think that this is a really exciting time to be asking those questions, and with the increasing amount of data coming available, we can really make rapid progress.
Awesome. Thank you. I love that you asked that question. What is normal and how much we need the individual differences, like how much we need to study these individual differences. And actually, I have a question for Monica about this. So what are the tools that can help us study these individual differences? And are there ways that we can measure this variability to move the needle on women's brain health?
Yeah, it's a great question. I think in the field of individual differences, as I mentioned, people are building models that predict individual differences from large data sets. And there's a really good recognition in the field that we need reliable brain data to do this. So this has led to pushes to collect sufficient resting state, fMRI data in order to build brain features that we can use to reliably predict behavior. There's also good recognition that we need reliable behavior measures, right? So there's work being done on questionnaires, tasks, making sure that the outcome measure we want to predict is reliable. But I think there's still more to be done in recognizing that even if we have a really reliable brain measure and a really reliable behavior measure, it's still just a snapshot at a single point in time. And that reliable measure could reflect something relatively stable about the person, but that doesn't mean every day of their life, every moment of the day, like that's their underlying ability. And so I say we need to be careful in thinking about these measures as like crystal ball that gives us insight into, exactly somebody's capacity. And so I'll use attention as an example. My lab studies attention, and we can have a really reliable measure of attention and a really reliable measure of the brain, and predict that pretty well. But other work in the lab has shown that we can use the same kind of predictive models to predict individual differences in how much sleep somebody gets. And so we need to know, right, like when we're predicting attention, are we just predicting who's slept badly or well, over the course of the last month, the last several months, the last year? We know things like sleep are affected by stress. Maybe they're affected by, right, if you're going through hormonal changes, if you are at a particular point in the menstrual cycle. And so I think we need to think about like what could be affecting both our brain and behavior measures to avoid falling into the trap of just thinking, right, like this is telling us exactly your underlying abilities or capacities. And so I think my concrete thought is we need to measure more sort of lifestyle experience variables, maybe very specifically, like what phase of a menstrual cycle is somebody in when we collect their data. That is not something that's often collected in big data sets, but that could influence brain and behavior in a way that we're overlooking. And so just taking into account these other factors when we build these individual difference models, I think is important. And we're sort of working on that in the lab now, particularly this idea of, can we build a model to predict phase in menstrual cycle? And if we take that into account, can we then improve models that predict behavior? By accounting for this variability that we're currently glossing over in many data sets.
Oh, I love that. Very cool. Yeah, so this next question is for Catie, and it actually kind of bookends nicely with what Monica was just talking about. What do you see as the biggest barrier for functional MRI to help us understand women's brains and to provide better care for people and women who have neurological and mental health disorders that are specific to women? And how do you think we can overcome that?
Yeah, so building on all the ideas that have been mentioned, I mean, one limitation of fMRI is that you are tied to the MRI scanner. And so our ability to sample from an individual is already quite limited, and it's kind of an unnatural environment. And so, that's kind of a fundamental limitation of fMRI. But I think that the efforts to acquire like longer duration data from individuals or more densely sampled across different timescales to be able to, leverage in order to build more powerful models is such a rich direction. And another one I would say is that the signals we measure from fMRI are very complex, and that they're indirect measures of the neural activity. And so our blood oxygen fMRI signals, they not only reflect the neural activity in a region, but also characteristics of the blood vessels and the blood flow responses, as well as more global physiological states like someone's blood pressure. And these might be quite important, I can imagine, in studying endocrine changes, and especially, across the lifespan. And so again, well, I think this raises some interesting questions in terms of modeling. It's also perhaps an opportunity that we can maybe, if we are able to sample the different amounts of variability and connect measures of physiology and of the body to the activity that we're seeing in the brain, then actually that could give us maybe some new insight into women's brain health. And so I guess following up on that, multimodal imaging, I think will be really powerful. So not only collecting fMRI, but collecting ASL, blood flow, perfusion measures, as well as dynamic measures of physiology from the body, your cardiac or your respiratory activity may help us not only kind of control for these as like potential confounds, but actually give us a lot more insight into the connections between these brain and physiological changes that are happening.
Oh, that was a great answer. Okay. I just want to remind people before we move on to the next question, if you have questions, you can enter them into the Q&A. We will monitor that. But yeah, okay, go ahead, Nina.
I think the next question is for me. I can just ask myself about it. It's about the advances in AI. So what advances in AI can be applied to brain imaging data? So there are lots of advances in AI. AI is a field that publishes a lot. So we're putting as a community 5,000 articles on arXiv every month. So to put that in context, since the 90s, the neuroimaging communities has put 50,000 published papers online. And in AI, we do that in 10 months. So we publish the equivalent of what brain imaging publishes, but has ever published, but in 10 months. So yeah, there are a lot of ideas that we can take from the AI community and bring it to the neuroimaging space. But one of the biggest problem is that the scale of the data set is just not the same. So in AI, we'll need tens of millions of images for a method to work. And as we said earlier, we have in the tens of thousands at best. These are the biggest neuroimaging data sets in the world that have only, quote unquote, 10,000 of images. So how can we, address that problem? So one thing that Catie just mentioned, which I think is very relevant, is to use multimodal data. So, okay, maybe we only quote unquote for AI have tens of thousand brain images, but maybe we can enrich that with other types of data. And then that can help the algorithm detect patterns. But even staying with imaging itself, there are new AI techniques that are designed to learn more from less. And there is a wide range of it. One example that I like is called equivariant deep learning. And the idea goes as follows. By default, an AI algorithm doesn't know what is an object and absolutely doesn't know what is a brain, in the sense that if you first show it one image of a brain and then show a second image, which is the same brain but rotated, from the AI perspective, it's a brand new thing, never seen that brain before. Just because an image is a table of numbers, you rotate the brain, the numbers all scramble up, and the AI has no idea what it is. And so for the AI to understand what is a brain, you need to show it so many brains in different orientation with different intensities, and all of these variations that are not physiologically relevant. And so in equivalent deep learning and these types of techniques, you build AI models in the way that they know what is an object, and you don't have to show 10 times or 100 times brain just for it to notice, oh, that's a rotation. That's one example of how AI can help neuroimaging.
Awesome. Okay. Yeah, and I think the sort of DeepSeek development has like pointed to how you can do things with less, right? So there are ways that you can design algorithms and sort of generative algorithms that you could generate data. So there's lots of different ways that you can overcome that lack of data. But you do need some data to start with, I guess. So
Exactly.
yeah, as long as we get some data and as much as we can, that would be great. All right. So the sort of next question, this is a question about PET imaging. It was originally for Joshua. Ted, do you know enough about PET imaging? Okay. Anyone PET imaging? No, I am aware that there is a PET tracer for estrogen receptors that is being used. And I think there's a couple of papers out of Weill Cornell from Lisa Mosconi's group looking at using a PET tracer that binds to estrogen. So I really think that that could be something potentially to look at sort of inter-individual variability in estrogen receptor availability and how it changes over menopause. So there's a preprint out there if people want to take a look, but I believe it's only being used at a couple of centers at the moment. So I think that that could be something that is to come to look at sort of estrogen receptors and how that changes brain activity and structure and function. Okay, so that was my attempt to answer the question about PET imaging. But I think the next one is good for everyone. So this is precision imaging has emerged as a promising area for human brain mapping in the last few years with really a lot of interest now in collecting data over time for, multiple time points in the same person as they go across their life for brain structure and organization and function. So what are the best practices and potential pitfalls that we should be aware of when we're designing this small n and extended sampling data? So anybody you want to answer that one?
I'll just put in a plug. So I think it's extremely important, right? Because then we can model within subject variability, which will allow us to ask right questions about changes within a person relating to changes in brain activity, which will then inform individual differences models, future trajectory models. But I think one thing that I want to plug is the importance of collecting data in different states or contexts. So not just, a lot of resting state data, every day for years, that would be amazing, but also we could collect task data, different kinds of tasks, different kinds of movies, stories, music, to really sample cognitive processes. If we're focusing on functional imaging in different contexts, because we know that if we're looking at brain networks, the configuration of the nodes in the network is changing depending on context. And there's predictive power for different outcomes is better when data are collected in some contexts versus others. And so I would just advocate collecting a wide variety of contexts instead of sort of going all in on one data collection type.
Does anyone have any other, do you want to?
One thought I might add is as we go to smaller N studies, I think that the quality of the data and how we denoise it also really, really matters. And so one emerging technique in fMRI acquisition is known as multi-echo imaging. And this has proven very powerful for precision studies in that it's a technique that allows you to try to parse different components within the bold signal and separate effects like head motion from what we think are more neurally driven effects and can maybe help to boost the sensitivity.
I just want to add to something that Monica said and that Catie said earlier, actually, which is that if we care about relating these precise measures of brain, which we made so much progress on recently, to behavior, we also need to focus on the behavior measurements too. So typically in studies, when we assess mood, we I'll ask kids in our studies like, oh, how have you felt over the past week? And I'm a pretty functional adult. I cannot remember how I felt over the last week on average. And so being able to get better measures of mood and psychiatric symptoms, I think is right now the measures we have are placing a real ceiling on how good we can do at prediction because they're not very reliable. They're not we're just missing a lot of signal. And so I think we need to, just like kind of the technical advancement we've had in imaging, we also need to engage people and from other fields who are having kind of advancement in behavioral measures too.
Like using smartphone surveys that you send out sort of on a daily or semi-daily basis. Yeah, I think there was a study that was focused on sort of looking at mood across women's cycles. It was done at Weill Cornell Medicine, and they had basically, I think it was like multiple times a day, they had a survey they had to fill out, and then they have this, this sampling of the mood in a very like high resolution way, which I'm sure is more reliable than individual things about saying like what you did over the last week, which again, I also don't know. Yeah. Well, more now I have an understanding of my baseline level, but it's fine. And I think Monica touched on this one. Do you think it's necessary to collect information regarding the timing of menstrual cycle for female participants if they have one when collecting neuroimaging? And do you see adjusting for this as becoming the best practice in the future? I think you touched on that, but I'd love to hear more about that.
I think it's important to collect. I think it won't take that long, especially with the standardized questionnaire that you're working on. So thank you. That would be extremely useful
Do you rely on self-report? That's my question. Do you rely on self-report or do you rely on testing, like the strips or anything?
That's a great question. So we haven't collected these data ourselves. We're working with open data sets at this point, but I'm curious to hear people's suggestions about best practices because I think the more information collected, the better, at least with one of the open data sets that we're working with. There's self-report and there's a flag for like hormonal birth control versus not, but there's different kinds of hormonal birth control and we're like missing some information there. So I think the data are important to collect. It's not clear whether at this point it's best to control included as a control variable to build different sorts of models. I think that is to be seen, but I don't think there's any downside in collecting the data.
Mm-hmm. Yeah, I will say that I have looked at the distribution of values for the Human Connectome Project young adult data when they ask when was your last period, and it's Gaussian distributed around 2 weeks before. So it can't be like it should be uniform. You know what I mean? So I think there's some, there's probably bias in the self-report. So I wonder if there's some, room here for perhaps like an app where you monitor over time, but like before the scan, so you know exactly when in the cycle that the person was. So yeah, maybe these things, because like we were just talking about, self-report can be very variable, it can be hard to do. So having these like outside measures that are more reliable might be helpful in that respect.
Absolutely. I'll just add one sentence, which is related to a question in the Q&A, which is the potential downside of standardizing across labs. So I'll put a plug. I think there could be value in collecting self-report and objective hormone measures and asking if models that relate brain to these outcome measures generalize across the two, because then you can get a sense of how much they're measuring the same thing. And if they don't, that tells you you're measuring different things. So I think there is a lot of value in collecting variable measures and not standardizing everything across all labs.
Yeah, and perhaps there's room for like a little bit of both, right? So you collect the standardized measure, which is kind of what people do now, right? There's the NIH toolbox where you have this sort of standardized measure of cognition, but you can also add your special like thing that you're excited about onto it on the side. Yeah.
And talking about hormonal cycles, actually, I think there's a question that's interesting that was added in the form, which is, do you think the findings on the structural changes incurred by hormonal cycles impact on brain atlases used regarding women subjects? We're starting to record this data more and more. Should we have different brain atlases for men and women, brain templates? What do you all think?
Well, I will say that, so I guess in my mind, I know there's a lot of work looking at how cortical thicknesses change, and I wouldn't necessarily say that should change the atlas that you analyze it using. However, I will plug in the idea of precision functional mapping, which is done on these, as we were talking about, these densely sampled longitudinal data sets where you can actually see an individual person's changes in regional functional connectivity, right? So right now, what we do is we take some population average atlas of, let's say, the functional networks from some set of people, and we co-register it into the person space, and then we calculate all of the connectivity between these regions. However, that person might not have the exact boundaries that exist in the atlas, right? And so a lot of people have been using these densely sampled longitudinal data to create an individual person's map, right? So you can actually see that there are changes in the individual person's map over time. And what I would love to see is somebody do this in a female data that is tracking someone along their menstrual cycle or along pregnancy or along menopause or even development, and looking at those individual level changes in the functional networks, and sort of how they relate to a person's feelings of well-being or whatever we're measuring outside of the scanner.
Yeah, so personalized atlases or personalized templates, maybe even at the individual level, which leads to the idea of these brain digital twins, which some people have started to build for, Alzheimer's, epilepsy, etc. And I think that's kind of the promise of what we can do with more data coming into our pipeline, which is a digital twin of your brain, which will be a replica of your brain, which is how your brain looks like if it was healthy. And then potentially you could use that digital twin to make a quantitative simulation on your own brain. So when I talk about this, I like to take the example of financial simulations. So, when you have to make a big life decision, for example, buying a house, you run through different simulation, like what if I take a loan, this loan over 20 years or that loan over 30 years, and you look how your financial health is going to evolve. But we don't have any quantitative simulations tool for our brain health. So what if you could have a brain digital twin in which you could run simulation? What if I take that birth control pill or this birth control pill, and how would that impact my brain? So I hope we're going to a future where the brain digital twin will become more common.
Great. All right, so we have another question from the audience. Which models and tools do you recommend to do correlations within these dense sampling data sets, considering we're dealing mostly with nonlinear, highly dynamic trajectories? So anyone want to answer that one? Ted?
I'm a huge fan of GAMs, generalized additive models.
I know you are. Yeah. No, they're very powerful. I agree. Simple, easy to use, and
I'm happy to geek out with anyone about GAMs anytime. We have a whole Slack channel in our lab about them. People like Bart Larson, who's also very involved in women's health stuff, are real experts, but happy to learn from them.
anyone else want to plug their own method? No? Okay. I'm not sure this question, I'm not sure I totally understand. In terms of predicting brain or behavior is more important. I'm not sure exactly. We were talking about sort of the mapping between the two, but depending on the modeling approach you use, you could either be doing one or the other, right? And I think linearly or mathematically speaking, it might not be that different between the two, predicting brain and behavior or behavior from brain, right? It's kind of trying to identify the relationship in this and which parts of the brain are mapping to which behavior. You can either use forward models or backward models, and yeah. I don't know if anyone has anything else to add. Okay, so we have another a little stickier one. So this one is about sort of.
Ted, do you have something to add?
Yeah, go please. Yes.
I would just say that, like, if we're going to be predicting behavior, like, we should always be asking up front, what is the reliability of the measure we're predicting? And if it's not reliable, we're just not going to do a good job with it, even if we have the most beautiful functional imaging in the world and like 1 million hours of fMRI. So just we often, as brain geeks, I'll speak for myself, I often skip that part, and it often bites us. So kind of knowing like the basics of the psychometrics of the measures you're predicting is really important.
Totally agree. And one other thing that might be interesting to keep in mind when thinking about making accurate predictions of behavior, we want a reliable behavior measure. There's also some evidence that using brain to predict brain to then predict behavior can improve predictions of behavior. So for example, imagine you only have resting state data, but you want to predict somebody's working memory abilities. Well, there's evidence that doing that from task fMRI data is more successful. So there are currently models where you can transform your rest data to what it would hypothetically look like if it had been collected during task, and then that in turn improves behavior predictions. And so there might be a sense in which, well, maybe we don't have all the personalized, really dense sample data that we want. Could we use the data we do have to project how we think this person's brain would look in a particular time or cognitive state to then improve behavior predictions?
Yeah, I love the idea of bootstrapping data, right? So like using what you have and trying to extract from it what you can get. And so we've been making tools in our lab to sort of estimate network disruption from lesions. So if you just have a clinical image where there's some kind of lesion or damage, and mapping that into some common tractography space where you can estimate structural connectivity and then feed it through another model that predicts estimated functional disruption. And it actually those estimates of like functional connectivity that are not at all based on resting state fMRI seem to do better than resting state fMRI at predicting some outcomes. So, there's also a lot of noise in fMRI, and I know a lot of us here have been working on trying to improve the SNR using sort of naturalistic stimuli, movie watching data, which is another thing that might be really interesting in the space of women's brain health because you can. Seems like the suggestion is that you get better SNR if you use some kind of naturalistic stimuli. So, yeah.
Yeah, but this notion of noise on the data, if you're trying to predict something, let's say behavior, and you know that your labels are noisy, so in a supervised learning setting, while you take an input, you want to predict an output, and your labels, your ground truth is noisy, there are dedicated machine learning methods that can take that into account, take into account that the ground truth is noisy. So you may want to look into that too. We have another question. Oh, moving away from more the technical discussion and moving into the socio-economical discussion. The question is, what kind of socio-economic considerations should researchers be aware of when recruiting women for studies and later generalizing findings? Any socio-economical considerations to be aware when recruiting women for studies?
I mean, I think one thing to keep in mind, there's a great paper written by a social psychologist, actually, who's my colleague, Lydia Emery, about recruiting people across different socioeconomic backgrounds. And it's, takes into account a lot of considerations for how to do this in social psychology. But I think as cognitive neuroscientists or human neuroscientists in general, we can use similar kinds of considerations. I mean, one thing that I think is very practical, like at our scan center, scanning during hours that are not just 9 to 5 in the middle of the day. I mean, taking into account that people may have other work obligations, childcare obligations, family care obligations, other things. And so being flexible in terms of like where and when we collect the data could make it more or less possible for women, at different stages of their lives to participate. And I think this is a like very concrete, practical consideration that we kind of sometimes just go, oh, here's the rules of the scan center, but advocating for maybe expanded hours, if that expands access, is important. This is like a really tiny piece of it, but one that's been very salient in my life recently.
Sure, thank you. All right, we have another question. Thanks everyone for the great session. My question is, do you think it would be possible to build a foundational brain model capable of predicting subsequent firing states or voltage levels, essentially simulating the brain? Or given its chaotic, dynamical nature, would data alone be insufficient, similar to the challenges faced in weather prediction? I can tell you that I've seen a foundational model of this, and it does seem to work pretty well. So there's David Van Dyke has a paper predicting sort of the next state of brain activity, and it seems to work pretty well. So I think it's doable. I've seen a lot of sort of monkey work where they record single neural data while the monkey is sort of performing a motor task, and the sort of next step prediction actually had the internal layers of this next step prediction have this beautiful sort of geometric structure. And so I think that it's totally possible in my mind.
Adding up on that, I've seen also a foundation brain model of mice from researchers here at UC Santa Barbara. And yeah, they were using different data modalities. So not only the voltage signals, but also what the mouse was seeing, the video of the mouse moving through the environment. And yeah, the predictions were pretty good. So now, obviously, this is in mice. How to do it in human brains is very more complicated. But from a technological perspective, yeah, we could do it.
Okay. The next one, I wanted to make sure we discuss this a bit, and I know there's a lot of uncertainty around this, but in light of recent political shifts, particularly in funding cuts to DEI-related initiatives, there's growing concern about the impact on women's health research. Could you discuss how these changes might affect the Anna Bowers Brain Health Initiative, as well as NIH-funded data sets like ABCD, which are also used to investigate women's health issues and sex differences? And then what can we researchers, both within and outside the US, do to safeguard and advance work in this critical area in this current and uncertain times? So I will say from my point of view, I'm continuing as if nothing has changed because I am operating under the optimistic hope that this is a blip. So the arc of history bends towards justice, but it's got like zigzags. You know what I'm saying? So we might be in a zig or a zag. So I am proceeding as much as I can, and of course I'm paying attention to the words that are being flagged in NIH studies, and I'm also paying attention to the fact that the sex as a biological variable from the NIH website disappeared. And so I think that those things can disappear, but if we as a community work together to keep it as something that we want to have as important, so if you're reviewing a paper, if you're reviewing a grant, if you're doing these things that are putting you in a position where the decisions are being made, keeping these things in mind and making them a priority, I think are something that we can do on our own. So it's my 2 cents.
Yeah, I agree with that. I think additionally, maybe it's time for scientists, especially scientists working in women's health, to speak up even more on how important these issues are and how underfunded and understudied they are. Because every time you talk about like the numbers, for example, out of the 50,000 brain imaging articles ever published, only less than half of 1% study pregnancy, menopause, menstrual cycles, so women's health factors. You give the statistic, people are going to listen. And this is an issue that transcends political perspective, as Emily was saying at the beginning of our session. So I think it's on us to bring this statistic to everybody so that everybody can recognize how big of an issue this is and how we should work together to address this.
Yeah, I just want to add that, like, even if it's from a non-political perspective, if you're not paying attention to sex differences and lifespan transitions related to endocrine shifts and socioeconomic effects and associations, there's a ton of signal in these associations. Like if you're not paying attention, you're just missing out. You're not going to model your data right. You're not going to get the right answer. So whatever your political stripes are, it's just what the data says. So we should follow the data as usual.
Yeah, so if Catie or Monica, if you have anything to add, if not, we can move. Let's see. I think there's one more question here. I'm not sure if anybody is aware of this one, but one of the WBHI seminars had a presenter that suggested when testing for interactions with sex, the p-value can be less stringent. I wonder if there are any formal guidelines or papers on this that the panel know about, or could they comment? I don't know if I know the exact person that you're talking about. It might have been Adrian Beltz, but maybe if somebody can add to that question, or if anybody has anything that they want to mention about sort of interaction terms with sex. Yeah, I think it was Adrian Beltz. So if you want to go back through and sort of identify her papers, that probably would be a good place to start. Yeah. And then I think we have one last question that's for everyone. So what would you suggest to someone who has expertise in computational imaging and wants to work on women's health, or who has expertise in women's health but wants to apply more advanced computational approaches? I like this bridging question as the last one. Anyone want to tackle that? Ted, do you want to do you have a
I mean,
suggestion?
I think it depends what career stage you're at. I think if you want to do that and you're a tenured faculty member, by all means, go ahead. If you're a trainee, I would find someone who's like been in the field and is really excited about it. I mean, the people on this call are there, but there's also awesome people who are just like Sheila Shanmugam is just starting her lab. That's like literally the focus of what she's doing. So I think that finding people who are really invested in training the next generation of people and bringing these two fields together, it would be awesome.
Yeah, I would say it's very good idea to bridge these fields and beyond also computational imaging, also going to machine learning and AI, because in machine learning and AI, there are so many researchers, right? It's a very hype and hot topic. So a lot of people working on that at the speed of light, but not necessarily aware of healthcare question or medical research question. So going to these conferences with like women's health question and trying to involve the machine learning and AI community to study real world problem, I think is very important. So yeah, by all means, go out there and try to reach the communities.
Yeah, I completely agree that the collaboration is so important. And I think in terms of this question, like on both sides for researchers in women's brain health, seeking computational or seeking to use computational techniques, figuring out what approaches are out there, the pace of the field is moving so quickly. And also, I think that researchers on the computational side will be really interested to know about the problems that the scientific questions that people are trying to address to study women's brain health, which would perhaps fuel new innovations in computation.
I totally agree with what Catie said. And just to add one more thing, if you're interested in moving into the space from computational neuro to looking at women's brain health or vice versa, also bring with you the questions that you are already expert on or you are really curious about, because that's how new questions, new ideas get injected into these fields. If we just keep trying to replicate what people have done, we get stuck in a little bit of cycle. But you say, I really care about chronotypes and how people are affected by, times of day against their chronotype. Well, maybe that's an interesting question to ask, how that interacts with, different stages of life, different stages of menstrual cycle. I mean, that's just one example. But bring with you those new exciting questions and realize that you have something to contribute to the field too, no matter which direction you're.
Okay, and then a selfish plug. I did organize this panel discussion, so I'll just take a few minutes. So Nina and I have been working with Women in Data Science, the WIDS group, as well as Kaggle, and we've been and also the HBN, so the Healthy Brain Network, and we've been putting together a data challenge, and it's out there now. You can go and enter. There is a little prize pot if you win. But essentially, we've been trying to figure out ways to get women and data scientists involved in women's health. And so this challenge they have every year involves, I think, about 5,000 people across the world, and they have to have at least half women on their teams. And so there is actually an active chat board where you can go and hook up with someone who wants to work on this project together. So you might have a background in computational neuroimaging and no knowledge of women's health, or you might have women's health and no computational background. And it's a good place for people to sort of connect and work on a project that's related to women's brain health. And this year, we're using the HBN data to look at predicting ADHD diagnosis from brain networks. And I hope in the future to be highlighting WBHI data, which is more sort of related to specifically to women's health. ADHD, while it is a sex sort of imbalanced diagnosis, it's a little bit different in the symptomology that present in women and men. It isn't something that's specific to women's health. So I would love to look at menstrual cycles or pregnancy or menopause once we have that data. Yeah, so please, I think Jaren put in the chat both the link to the survey, so please fill that out about our seminar series, as well as the link to the WIDS Datathon. Yeah, and so I think this is actually a good question. What is your take on studies that look at the effects of menopause on the brain but do not control for chronological aging? So, for example, they don't age match and compare older postmenopausal women to younger premenopausal women. So this is something that I've been actively working on. And I've taken multiple approaches to solving this. And one of them is to perhaps use males that are aging as a like outside map of aging. Of course, it's not perfect because men and women are different. Hello. But using that as sort of a way to get an aging trajectory is one way that we've thought about doing this. If anyone else has anything they want to discuss, that would be great. It's a hard problem though, because of course you can't. Someone who is postmenopausal but younger is going to be sort of a biased data point, right? So they went through menopause early, perhaps for some underlying health reason. So just taking people who go through menopause early and people who go through menopause later and trying to age match that's a little bit fraught as well. So there's lots of these sort of problems. So that's why I think we should do longitudinal data collection of the same person over time, right? Like that's the optimal thing. Yeah.
Yeah, I think that debate has been a huge debate in how to study puberty also, and people
Yeah.
have resolved it and have largely come to we need to follow people longitudinally. So totally agree with you.
Yeah. There you go. Good. Okay. Yeah. Any parting words? Encouragement? Keep your chin up. Like I said, arcs towards justice, but also zigzags. So just everybody hold on. One other question. I'll get this last one. Should we start considering menopause symptoms and their severity? I think a resounding yes, 100%. Because I think as Ted mentioned earlier, there is this like wide variability in the level of symptomology that people have. The level of, how I react to my own hormone fluctuations is very different from how someone else reacts to their hormone fluctuations. Just look at pregnancy as an example. Some people have terrible morning sickness for the entirety of their pregnancy. Other people have nothing. So it's very variable, and it can even vary between pregnancies. So there's a very big wide range about how people react to hormones. So I think definitely looking at symptomology and severity would be something that would be very interesting to investigate. Okay.
Yeah, I completely agree. All right, I think we're nearing the end of this panel. By the way, we wanted to say that it's been a year that we're organizing this seminar, and we would like to know how well we're doing. So Amy and Sharon have put a link for a feedback form that you can click on and let us know how we're doing. Also, would like to thank Sharon for all the work she's been doing, not only to organize this panel, but like the whole seminar series. So thank you.
Yeah, thank you. And thank you to all of our panel members for joining us today. It was a really great discussion, and I hope people have taken some nuggets away from that to apply them in their own work. And again, please sign up for the data challenge if you haven't already. It's a great opportunity to get started on using your computational skills in women's brain health or vice versa. So thank you.
Thank you.
Thank you.
Thank you.
Thank you very much for your participation. It was a great call. Bye-bye.
Bye-bye.
Bye.